So this is a bit of an intro about DNA evidence before we launch into how we're actually putting that into our family history programs, such as Danielle was just describing. I'd like to just confirm that this is my idea about DNA evidence. There's no law about it, and some of the things I say you can and can't do is just my own rules, I suppose. It's really up to you to work with what you've got and just consider some of the things that I'm going to talk about today. Okay, so this is about using DNA evidence to confirm your ancestors. So I just want to talk about what is DNA evidence. What are we talking about when we're talking about putting DNA evidence into our family trees? And just as a first point, we need to always remember that DNA rarely proves a relationship. You hear me talk about DNA confirmation, whether an ancestor is confirmed by DNA or not confirmed by DNA, or whether we're not quite sure and it's a tentative relationship. So it's all about gathering evidence, and just like traditional genealogy, we'd like to gather lots of evidence to confirm our ancestors. Now, isn't a DNA match sufficient evidence? We just need to remember that not all DNA matches are the same, and that there are different types of DNA evidence, different things that we might like to consider when we're looking at confirming our ancestors. So first of all, we have matches. But matches aren't all the same. So things you might like to consider are they within the shared centimorgan range for the predicted relationship? Do you have shared match clusters or triangulated groups? Are they third cousins or are they a distant match? Is there a paper trail or isn't there? You might like to use Watto charts as evidence, or Watto charts are from DNA Painter. Looking at probabilities of relationships, you might like to quote those as evidence. Ethnicity reports, and of course Y-DNA and mtDNA are particularly useful for more distant matches. But are they clues or evidence? These are some of the things we just need to think about. And in terms of the standards of evidence, we have the genealogical proof standard, which has been around for a long time. And it's got those 5 points there that anyone who's been involved in traditional research will be well aware of in terms of doing their own genealogy. In about 2019, they brought in some DNA evidence standards, and in my opinion, and in many people's opinion, the point about reasonably exhaustive research suggests that you can't actually meet that test if you haven't considered DNA evidence, if that's possible. Obviously, in some cases, there's no testers, etc. But most cases, you can actually get DNA evidence, and we really need to incorporate that into our research, in any research that we're doing about whether we're looking at the right ancestors and confirming the paper records. Now, under the standards in the GPS for DNA evidence, there's a whole list of different things we need to consider. And some of those we'll be talking about today, but this presentation is really about the second one, and analysing DNA test results and what sort of things you might be analysing. And under that particular point in the GPS, there's a whole lot of other points. So I'm going to talk about some of these today as we work through the session, but mostly the earlier ones. But all of those things need to be taken into account when you're analysing DNA results. So you've got to really, it's not just about getting a match. So are we looking at clues or evidence? Something we constantly talk about. And you'll all probably, most of you, I imagine, would recognise this slide that Christine Woodlands developed back in 2020 about our DNA questions. And in 80% of the time, we can actually work out the answers to our DNA questions using tree triangulation and total centimorgans approach. But for more distant ancestors, we need to be looking at segment triangulation and chromosome analysis and other tools to confirm our ancestors. So if taking that information and applying it to DNA evidence, we really have the same sort of thing, that we have one approach for our third cousins and closer, and probably a different approach for, or a more extensive approach for, confirming more distant relationships. So on that red left-hand side, tree triangulation and total centimorgans are pretty good if you've got them meeting a predicted, you know who the most recent common ancestor is, they are in the right centimorgan range, and you've got a corresponding paper trail, you're on pretty good ground. But if there's no paper trail, even with third cousins and closer, if you've identified a most recent common ancestor in that range, but if there's no paper trail, there's missing gaps, you need to actually support that with other supporting evidence and using tools like Watto, cluster analysis to develop your hypothesis. Over on the right-hand side, for your more distant relationships, the total shared centimorgans becomes less important, as we know, because of recombination and the strange things that can happen with recombination, in that not everyone gets the same amount of DNA from those ancestors. And so you need to do a lot more work in terms of looking at other people's tests and that sort of thing to confirm evidence of a shared ancestor. So, and again, with no paper trail, you need to be getting a lot more evidence together, using multiple tests, using multiple triangulated groups, and also probably Y and MT, where you can find a tester that might help you there, or if you've got other ways to confirm that through other people and other groups. So again, you're developing your hypothesis all the time. And even in the shared centimorgans, you know, very few relationships actually confirm an ancestor. Even like parent-child and siblings, full and half siblings, they usually come up with, if you're looking at the match in, say, Ancestry, they'll have 100% as the probability. But even with those relationships, even though they say 100%, you've still got to consider the issue of identical twins and that sort of thing. So nothing is certain. And that's why you need to be continually looking at gathering additional evidence to prove your hypothesis, particularly with those cases where there's no paper trail or gaps in the paper trail. So as far as we go in terms of researching, traditional research, we've always worked from the premise of working backwards in time, from the present to the past, verifying generations, looking at all the sources, as our process. And genetic research is really exactly the same. You've got to DNA confirm your closest relatives before you can start moving back in the generations. And ideally, you're striving to find the segments that belong to your closest ancestors, so you can build on those segments and develop your pedigree base from a genetic point of view, that all your ancestors are DNA confirmed. But not all the genetic sources are the same, so you've got to be continually looking at what you're looking at. Is it evidence, or is it just a clue for me and I need to do more work? And some of the, additional work that you might do is looking at, as we already talked about, shared centimorgans and probabilities, walking back the chromosomes, ethnicity reports, and going backwards and forwards between the genetic evidence and the paper records all the time. So what's the genetic evidence telling you? Go back to the paper records, see if you can tie the two in. And the GPS talks about resolving conflicting evidence, and probably in the DNA world, it's looking at where you've got low probabilities for the relationship. Why is it so? Continuing to question those things, looking at whether you've got possibly false positive triangulated groups, where pile-up regions are. So are they really matching? So is it a false match or is it a real match? And that's probably more important at the distant relationships. So we've got the same concepts happening, and our research techniques haven't really changed. And we're just talking about DNA as another type of evidence that you have to sort of try and demonstrate in reports or anything that you're doing in that regard. So the confirmation process, in my opinion, is work on your biggest matches first, identify those high centimorgan matches and where you've got shared paper trail. And you're using that information to make sure your pedigree base is solid, that you know that those earlier ancestors are your ancestors. And then you've got to try and get the segment data if you want to push back past second great grandparents. it's not so important for the confirmation side of things, but if you want to actually push back your pedigree, which is what I'm trying to do, I'm trying to get back to my 5th great grandparents and have all lines DNA confirmed. And to do that, you need to actually start looking at the segment data. But you can't do that until you've actually done all your close ancestors. And for that, I talk about up to 2nd great grandparents. Then use clustering and triangulation techniques to push them back further generations. And as I said, in my opinion, shared segments are the only evidence for past 2nd great grandparents, but that's just my rule. But I'll talk a little bit more about that as we move forward. So the first point in the GPS is about accuracy, completeness, and depth of pedigrees. And you may have seen this chart before, which is from DNA Painter, which is showing tree completeness. So you can put in there how many ancestors you've identified, and it'll give you this little chart for showing you how you're going in filling out your tree. And you can see from the image at the back there that there's a fairly well developed tree, but there are some gaps where the grey pieces are. So this particular tree has 164 out of 254 ancestors identified, which is 61%. But what that means is that there's 39% of this particular pedigree's ancestors who we don't know who they are. And without both you and your match having complete pedigrees, you won't know if there are more common ancestors. And point 2 of the GPS is that you need to have explored whether there is a possibility of more than one shared ancestors between 2 testers. And the further you go back, the more chance of this is happening. Because you may be related to your matches in multiple ways. And the classic example of that is, the story about Charlemagne, who they reckon, I don't know what the percentage is, but a very high percentage of Western Europe can trace their ancestry back to Charlemagne. But if you do the sums, each generation we double our ancestors. So I was talking there about 254. So the next generation is what's that, 508. If you keep doing the multiplication of generations, we can't all have unique pedigrees because we've got far too many people on the planet than there were at the time of Charlemagne. So there's definitely a lot of multiple shared ancestors that we would have with other people going back further. So we need to make sure that we've got the right ancestor, and that's where segment triangulation comes in. And the other thing you need to remember is that your genealogical tree is different to your genetic tree. This is the porcupine chart that was developed by Jim Bartlett, and it's showing down the bottom there in the solid area, 1st great grandparents who are your 2nd cousins. 99 to 100% of the time you're going to match those people. But as you work back, 3rd cousins, it starts dropping away. You're down to 90% of the time you might match a person. And at 6 cousins, there's only a 2 to 11% chance of matching. So it's important to test as many relatives as you can, and pretty much essential for trying to confirm your more distant ancestors back past 3rd cousins, because you may not have got the DNA from that particular ancestor. So you need to find someone who did and to be able to get back further. So multiple testings and reporting about not just your results, but other people's results in your evidence about confirming a particular ancestor becomes important. So as far as DNA confirmations go, when we first start out in family history, there's a few parallels. We're always told, don't rely on the index, check the original. So in genetic genealogy, it's the same. Total centimorgans and shared matches, they're clues. They give us a lot of information, and most of the time it's pretty reliable. But segment triangulation is really the gold standard, in my opinion, and is actually evidence. And to me, that's sort of like, a primary record, and total centimorgans is more like a secondary source. So it's building on your evidence, but it's still margin for error. So And this is a chart that came from DNA Painter, and just sort of gives a bit of an indication about what you can DNA confirm. And again, this is my theories. So the dark green there is about individual ancestors, and the light green is talking about confirming a couple. So in my opinion, you can only confirm individual ancestors up to second great grandparents with total shared centimorgans and a paper trail. When you get out to these ancestral couples at the second great grandparent level, you really need to be looking at, you may have matches to those people, but then going back another generation, you need to make sure you've got matches on both sides. And, you know, third cousins, they can share different ranges. And I think my smallest one is 4 segments, and my largest one is about 8 segments. So those 4 segments, if they're all coming from P1 and not P2, then you've only really confirmed one side of that particular couple. So you're constantly looking at those segments there to try and actually get them back on both sides before you can actually confirm those second great grandparents around the outer ring there as confirmed, and give them the darker green color if you're trying to display those things in a chart like this. So beyond third cousins, or where there's no paper trail, the same thing applies. You need to be getting back to segment data, and many shared matches will be distant cousins, possibly matching on those unknown lines. And you need to be careful when you're dealing with matches less than 30 centimorgans, because they can be some of those segments that have hung on for a long time. I recently discovered a triangulated group that went back, I think it was, I can't think now what it was, but it was like about an 8th cousin or something, but I'm pretty confident that it's confirmed. But it was a 20 centimorgan segment, which you know, sometimes you just can't find them, but they can be hanging on from a long way back. So you just need to be mindful of that. DNA confirmed past third cousins, in my opinion, again, really segment triangulation is required to identify the most recent common ancestor. I'm not going to go through segment triangulation. That's a whole session in itself, but essentially you need to have people who are matching on the same chromosome in the same segment area, and going back through tree triangulation to the same ancestor or ancestral couple, and coming through different descent lines so that the matches aren't all coming from the same person. And you can see in this example here, in the segment I have up the top here with, for the great nephew, the blue segment there, this is actually showing a triangulated group with my mother. And the great nephew, he shares her parents because he's related via her sister. So that blue segment, that blue segment could be a mix of mum's parents, so it could be maternal or paternal. But if you work through all these other people, going down to this third cousin twice removed, it's a much smaller segment, but that much smaller segment is coming from those great grandparents, and whatever the, you know, whoever they are, maternal or paternal, you can confirm that amount of the segment, not the whole blue one, just that little small bit of green there. So that's segment triangulation. But I'll be running a course later in the year about segment triangle, chromosome analysis. So if you want to learn more about that, you come along to those. Okay, so the next I just wanted to mention briefly was about tentative versus confirmed. As I said before, DNA rarely proves a relationship, and you need to be building on your hypothesis all the time, going back and forth between the paper records. And these are just some points that I just wanted to talk about in terms of gathering more evidence for your conclusions. So the first one there, under third cousin and closest, if you've got the assess the shared centimorgans for a relationship and they're good, you might just want to give it a big tick and say, right, just don't need to do anything more with that match. But really, they are the key to getting further and further back, because what you next need to do is to be looking at shared match groups that that particular person might have, so you know where they fit. Who are all the people that they share with, and can you find some more common ancestors with those people? And then try and get their segment data, because if you want to build back with your on your pedigree base, your DNA pedigree base, or your genetic pedigree base, you really need that data to move forward. And these cousins are the best ones to start with, so that you can identify triangulated groups. You might be wanting to use some Watto analysis to use as evidence, but remember, Watto only is best with larger matches over 40 centimorgans, and ethnicity reports might be another one you might be wanting to use as evidence to prove or confirm a relationship. Higher than second great grandparents, more distant and third cousins, similar to what I've been saying before, really you need to be interrogating those segments of those closer cousins and identifying triangulated groups and walking back the chromosome over time. Now, Watto at this level becomes pretty much, it's not really that useful other than as a visual. And I'll just show you a couple examples of how I use them as visuals, and you might like to use those charts as evidence in your conclusions. There's also cluster reports, ethnicity reports, etc. So always encourage your ancestry DNA matches to upload to other sites. And most importantly, do the genealogy to continually be looking through all of this information and linking it back to the paper records. And really use multiple kits to verify your findings. Always try and test close cousins, and, you know, try and get, if they're on Ancestry, look at see if you can get access to their results, so that you can look at how they're matching with the same people. And the same thing for the more distant ancestors, getting people on GEDmatch so that you can run reports between different people and compare notes. You don't have to have tested everybody that you use as evidence for your close cousins, particularly if they're on GEDmatch. If you can get them on GEDmatch, you can do the analysis from there. So that's really useful. Again, warning, where there's no paper trail, you've really got to do a lot more work. So these are all ideas about how you can actually gather more of that corroborating evidence to confirm your hypothesis. So I just wanted to say what is not DNA confirmed before we move forward. And we're all getting these common ancestors. They're wonderful, and they're very good clues for your genealogical research. But sharing ancestors is not necessarily proof of genetic relationship. And I just want to talk through this one. Some of you may have seen me talk about this before. The problem with this particular match is that half the pedigree's not there. He's only got a maternal side. So could there be some other ancestors that are at play here? So genealogically, it's correct. It goes back. It was on the same line as what I thought was on my mother's paternal side. But when you look at her paternal side, it's going back 7 generations to the 5th great grandparents. And I only have matches back to her grandfather. So this is the first match I have on this line. So it's ringing danger bells to me. You've really got to be getting matches in at this level and moving back. You can't just go up to a confirmed cousin at 6th cousin level. You really need to be making sure that the lower generations are all DNA confirmed. So that's the problem with this one. And when you look at this match, it's 15 centimorgans, but the shared centimorgan project, it's a 60% chance of being right, but it's got all these other relationships that could be possible. So you really need to be thinking about whether that's correct. So I don't think a single match in isolation back that far can be considered DNA confirmed. But it is useful for genealogical research, and often, you know, you may have a genealogical connection with someone. It might not be a genetic connection, but it is very handy for your further research, and you might pick up on some lines that you didn't realize existed. So always explore them. And it is a match and it might be correct. So for something like that, I'd always be looking to try and get up to a chromosome site. But I'd add it to my tree, and I do add a lot of DNA matches to my tree, and I'd mark it tentative and add an appropriate source citation, which we'll talk about a bit later. Just so that it's in my tree and I don't forget about it, really. And don't forget, in terms of gathering evidence for your ancestors, Y-DNA and mtDNA for distant relationships, they're probably the best test you can get, really, back at the fourth great-grandparent or fifth great-grandparent level. But often it's difficult to find the right people. And don't invest in your tests for these unless you're pretty confident that your base is solid through your autosomal analysis. So make sure you've confirmed, at least a few generations. I'm pretty confident that you're actually your fourth great-grandparent is the one you're expecting. And if you haven't got any matches to your first great-grandparent, sort that out first before you go spending lots of money on tests, because you could be paying a lot of money for a test that a person who's not necessarily related to you, particularly if you're seeking somebody out to test for you. Different if you're testing your own line. So in terms of assessing likely relationships, we have the Shared Centimorgan project, but I like to look at this for the averages, just to see where people should fit. Remember, it's crowdsourced and the range is crowdsourced, so it's not 100% accurate. Ancestry DNA has a good tool where on a particular match, it'll show you the predictive relationships, and these are pretty good. You can also get that result from the DNA Painter Shared Centimorgan project by keying in the centimorgans, and looking at the probabilities. It's a similar thing. So always look at those percentages, because they can be very useful, because they can highlight somewhere you might not have thought of, like half second cousin 3 times removed. This doesn't come immediately to the brain, I don't think. But then when you see it there, you start to think about some of those other relationships that might be possible. And Watto, I've talked about Watto, and Watto charts are great evidence to be using in your analysis, and when doing reports and that sort of thing, or coming to conclusions about particular ancestors. But again, it's best used for larger matches where you've got over 40, because the stats don't work below 40 centimorgans. But for more distant and smaller matches, it can be very useful as a digital aid. So this is just a quick chart example of a chart that I put together for trying to work out where this match Anne fitted, and she matched my mum at 108 centimorgans. And I came up with a few hypotheses, but as you can see on this chart, they're all pretty small matches. And so Watto doesn't really work for probabilities that low, or for matches, shared centimorgans that low. So the chart's insufficient for drawing conclusions, but I like to put some of these things in a PowerPoint presentation as I'm working with a match, and I write on here, so various things, and it helps me to come back to where I got up to last time. So I've got in here some various possibilities that I think she might fit, where she might fit, but it's very inconclusive. But again, you might want to use that chart as an evidence somewhere. What I would then do is then add to that PowerPoint, on top of an overlay on the Watto chart, details about triangulated groups that I've got. So I've got in here information at chromosome 2. I've now got a Pace-Parker segments, and different matches coming out here from Sarah Monk. So I might put this away and not look at it for 12 months, and it's a good way to get back and remember where I'm up to. So in this particular example, I'm still trying to work out where Anne fits in, but it's not very conclusive for Anne. but it's an increased confidence level because I've got some triangulated groups. So it's the additional evidence to the first chart that I had, which just had the ancestry matches in, or the total centimorgan matches in. And then if I go another step, looking at the same information for my own family, I might not be clear about where Anne fits in, but this chart can be useful for me saying, okay, the centimorgans that I share with Anne, even though I don't know exactly where she fits in, I'm pretty confident they all come from this Abigail Pace where the red box is. And the reason for that is that we've got here in the next generation up, Anne's got matches from both these couples, but they're two brothers marrying two sisters. So probably we share, at very least, we share this couple out here, but that's pretty tentative because we're not quite sure where she fits in. But for my analysis, doesn't really matter because I reckon it all comes from Abigail Pace, and that's what I would be concluding in terms of working with Anne's match for my own family history. Working for Anne to work out where she fits is a completely different question, but not something that I necessarily need to resolve. So some other supporting evidence you might want to use is clusters or shared matches and ethnicity estimates, that sort of thing. So what you need to be doing when you're gathering your evidence for your DNA confirmations, you continually need to be working on your pedigree. As I said earlier, identify those close unknown ancestors. You've got to be firm on your base before you start. Then the second priority would be working with anyone up to the second great grandparent level who hasn't got any DNA matches. You might have a problem there. And then the third priority, of course, is to be continually looking to build out your tree. And I'm trying to build out my tree to the 254 level. That's probably enough. I've got a lot of lines that are going back further generations, but in terms of DNA, I think that's probably the maximum that you might want to be doing through autosomal analysis. And then you confirm it with your genetic evidence, building a solid base, identifying and walking back the segments over time, and then working with other reports such as WATO and event ethnicity and all those sorts of things. And tying it back into the paper records is really important. So the genealogical proof standard applies to DNA analysis. Key message is don't DNA confirm because you've just got one match. But the genealogical proof standard also talks about complete and accurate source citations. So what are they and how do we record them? Some of the sites have actually come up with methods for recording DNA evidence, such as Ancestry. Ancestry has these thing called tree tags. You can see those there. They have common ancestor, DNA connection, DNA match. I don't actually think they go far enough, so I've created my own custom tags, and I have DNA confirmed to couple, DNA confirmed, DNA tentative. And so for a DNA confirmed, I would have someone like Abigail Pace, who I know is my ancestor. I've got matches back on both her sides, on her father's side and her mother's side. And so she's fully confirmed as an individual. Edward Roberts Baker, only have him confirmed a couple. I've got plenty of people who share him as an ancestor with his wife, Abigail Courtney, but we've only got matches back on his mother's side and not on his father's side. So, or we do have that one match I talked about earlier, which I'm very sus about. So he's only, he's confirmed, but he's confirmed at the couple level, not at the individual level. And then DNA tentative is his supposed father, Edward Roberts. Only that one match, nothing else. And I'm pretty confident with the tree of Edward Roberts is correct, and it's a very extensive tree. Goes way back on many of his lines. So why haven't I got any DNA matches? Have to start thinking that perhaps he might not be my ancestor. But these tags are useful for navigating in your tree, but they don't meet the GPS requirements of full and complete source citations. Same at DNA Painter. DNA Painter has a wonderful chart, which I use, and they have these little symbols here that you can add as being a genetic ancestor. But to me, they don't go far enough. Same reasons. I like to look at who's DNA confirmed as an individual. I use the color coding on the tree to differentiate between those people, as I sort of mentioned before on those round charts. And DNA confirmed show up as the dark green, ancestral couples, the lighter green, tentative as the orange, and somewhere where I've got no matches, like Arthur George Courtney, everyone's heard me talk about him before. He needs to be more research. So he's my priority, and then I'll probably work through the tentative matches, then looking at the DNA confirmed ancestral couples some more. So they're the 4 ancestors at that level that I'm still working on. Again, this is probably more useful, but it doesn't meet the GPS standard either. And images, sorry, not images. It's sometimes, you know, you've got tentative matches. Now, so for that Roberts example, I've only got the 1 match. But on other lines, I've got 10 and 20 matches. So colouring in the chart doesn't really give you the weight of evidence that you've got for those particular lines. So how do we manage that? So for years, I've been pondering this question and looking at how I can actually incorporate the outcomes of these analysis tools into my tree, because I'm constantly going back and forth between my DNA results and my pedigree, looking at and revisiting things that I'd done before. So it wasn't clear how many matches I had for each ancestor, whether I was confident, did I have segment data, did I have triangulation or any of those things. And more often than not, the main problem was I'd be looking, of course I add a lot of people to my tree for DNA matches and shared clusters and things like that. I had all these people in my tree and I was thinking to myself, who the hell are these people and why have I added them? So I looked at different ways to record some of this information and have it readily accessible as I was doing my research. So today's session is about ways to bring that evidence into our family history, and Danielle's going to talk about her method, and then I'm going to talk about what I've come up with in keeping track in your tree. So hopefully that's given you, if those of you who aren't sure what we're talking about when we're talking about DNA evidence, some ideas about what sort of things we're trying to incorporate into these, into our family history programs. Okay, so that's all from me. Thanks very much.